The nervous system is part of everything we do, but to summarize its functions, it receives, integrates, and controls. Namely, it receives sensory input, information about the external and internal environment. It then processes or integrates that information, analyzes it to make sense of it. This occurs in what's called the central nervous system, the brain, and the spinal cord. Then it sends signals to our effector organs, various types of muscles and glands, to trigger an appropriate response. This might remind you of homeostasis, where we had receptor, control center, and effector working together to maintain it. Pretty similar to receiving sensory input, and then the brain integrating that information, then effector organs being triggered. So the nervous system is integral to maintaining homeostasis, allowing us to be aware of changes in our environment and in our bodies, and then triggering changes that need to occur so conditions in our bodies remain normal and healthy. Lastly, the nervous system establishes and maintains all mental activity. So this includes spatial awareness, personality, learning and memory, everything that we rely on. The nervous system can broadly be divided into two branches: the central nervous system, or CNS, and the peripheral nervous system, or PNS. The central nervous system consists of the brain and spinal cord, so the organs that are central in the body. This is where integration, processing, and analysis takes place. The peripheral nervous system is everything else. So all the nerves that penetrate the extremities, the trunk, the face of the body, all the receptors that pick up on sensory information, both external and internal. Basically, if it's not part of the brain or the cord, it's going to be peripheral nervous system. The peripheral nervous system can further be subdivided into somatic, autonomic, and enteric nervous systems. All of these have both sensory and motor components. Sensory indicates information traveling towards the central nervous system from various sensory receptors. So, this is information about the internal and external environment being collected and then shuttled off to the central nervous system, where it can be analyzed. Motor is the opposite direction, from the central nervous system to our effectors, skeletal, cardiac, smooth muscle, and all the glands of the body. So these organs can make the necessary changes. Motor in a car makes it move, and the motor division is what's responsible for movement of the body. Somatic means conscious or voluntary. So this includes all conscious sensations as well as voluntary movement. Thinking back to chapter 7, you may be able to guess what the somatic effector organs are. They would be voluntary. Well, what type of muscle is voluntary? It's our skeletal muscle, right? So skeletal muscle would be the effector organ here. Autonomic sounds like automatic, and that's basically what it means. Unconscious information about conditions inside and outside ourselves. So things like glucose and calcium levels of the body, heart rate, blood pressure. And then the effector organs, the motor division, will trigger cardiac muscle, smooth muscle, and secretion from glands. When I was taking anatomy, this is all that we had to learn. But since then, we have a new division to be responsible for. That is the enteric nervous system, which has everything to do with digestion. So the sensory division here would keep tabs on if the GI tract is full, what it contains. The motor division would control movement of the GI tract, secretion of glands. So it's automatic too, or unconscious, but it is concerned only with digestion. So when you think enteric, just think, okay, digestive system, digestion. That's the only thing it's responsible for. This is a good visual of those divisions. We have the central nervous system, so our brain, our spinal cord. This is responsible for processing or analysis or integration. And then we have all sensation. Sensory division is just placed out here, so both conscious and unconscious is all lumped into one category. But we can see somatic and autonomic motor output, where somatic is responsible for voluntary conscious effector organs, whereas autonomic is responsible for unconscious, involuntary, automatic effector organs, cardiac, smooth muscle, glands. You can see here it does get more complicated. The autonomic nervous system is divided further into two divisions that maintain homeostasis under different conditions. The sympathetic division is in charge of our fight or flight response, or our stress response. Whenever we are stressed, the sympathetic division will activate our autonomic unconscious effector organs to respond in the appropriate manner. This is going to amplify our physical response. So heart rate goes up, blood pressure goes up, breathing rate goes up. Whereas the parasympathetic division is our rest and digest division. This is what controls those effector organs whenever everything is normal, everything is fine, we're not stressed. It's our Netflix and chill division, I suppose. So our heart rate relaxes, blood pressure, respiratory rate relaxes, more blood is going to flow to our digestive organs. Now, we already looked at nervous tissue in Chapter 4, and luckily for us there was just one type of tissue, which was really nice. after looking at all those different types of connective tissue and epithelium. However, that one type of tissue is a bit complicated because there are two types of cells that work to maintain it. The functional cells are the neurons or nerve cells, whereas the support cells are the neuroglia or glial cells. Neurons are the stars of the show, meaning that they are the ones that manipulate ions to conduct impulses, stimuli, or action potentials. So they are capable of receiving stimuli, processing and analyzing that input, and then transmitting signals to other cells. They are the movers and shakers of nervous tissue. They give nervous tissue and the whole nervous system its function. They work hard, so they require lots of oxygen. They are only capable of metabolizing glucose, so they require a lot of glucose sugar as well. The brain is going to use up about 20% of our glucose at any given time. Neurons do not divide after they mature, so any healing that occurs here will largely be a function of the neuroglia cells. Neurons have a characteristic structure that has three components: two types of processes, the dendrites and axon, and then the cell body. Dendrites are short, tapered processes. Many times there's multiple dendrites. They will receive a stimulus from another neuron or from sensory receptors, and then they will conduct that stimulus to the cell body. The cell body is this large part of the cell. It contains that large nucleus and other organelles like the ER, mitochondria, the Golgi. The cell body is responsible for processing the stimulus. Once it's processed, the stimulus will head to the axon hillock, which is a narrowed portion that leads then to the axon, which is a long, extended process. Usually there's one, though you can see it can branch. The axon is going to transmit that stimulus to another cell, be it a gland, a muscle, or another neuron. In order to interact with many different cells, many different dendrites or muscle fibers, the axon divides down here into many axon terminals or presynaptic terminals. This term might look familiar because this is one half of a synapse, like the neuromuscular junction. So the whole neuron does what the nervous system does, only in miniature. It receives input, processes that information, then transmits it down the line to another cell. Notice also there's a one-way flow of information from dendrite to cell body to axon. Structurally, there are three types of neurons. This is based on the number and arrangement of processes. Most neurons are multipolar. Multi for multiple. This means there are many processes, lots of dendrites, and a single axon. Multipolar neurons are found making up most of the central nervous system, the brain and cord, and also most motor neurons leading to our effector organs are multipolar. So that's pretty much all of the central nervous system and a good part of the peripheral nervous system. Because this type of neuron is the most common, whenever you see a diagram or model of a neuron, odds are it will be multipolar. So looking back here, this is a multipolar neuron. We see here it's a typical motor neuron. So that makes sense, right? As most motor neurons are multipolar. Bipolar neurons, bi for two, consist of two processes, one dendrite plus one axon. These are quite rare, being associated only with the special senses, which include vision, hearing, smell, taste, and equilibrium. So they will be found in the organs that detect these senses: the eye, the tongue, the nasal cavity, and the ear. And we can see it, just two poles or ends, a dendrite and an axon. Pseudo-unipolar or just plain unipolar neurons have just one process, uni for one, like a unicorn has one horn. So they have one axon and no dendrites. However, they are called pseudo-unipolar because they actually begin life as a bipolar neuron, but then the two processes fuse. And they also functionally do have two ends in that there is a sensory portion, you could think of it as a dendrite-like portion, and then an axon portion. So there's a part that receives info and a part that transmits the info along. These comprise all of our general sensory neurons. General senses are those that are felt all over the body with no special organs dedicated to them. So touch, pressure, pain, itch, tickle, thermal sensations would be our general senses, found all over the body, associated with the skin. Now, we can also divide neurons into functional classifications that overlap with these structural classifications. Those would be sensory neurons, then association, or sometimes called interneurons, and then motor neurons. Right, so there's some overlap in that sensory neurons are either bi or unipolar, depending on what sense. And then association or interneurons, these are neurons that are in between sensory and motor neurons that process and integrate information. They're going to be multipolar, found in the brain and the spinal cord. And motor neurons are also multipolar. Another term I'm going to summarize here, might as well, since we're kind of talking about it. There we go. You'll see other terms that mean sensory and motor. Sensory equals afferent, and motor equals efferent. These terms are used interchangeably, so depending on the resource you're looking at, sensory could be described as afferent, going towards the CNS, Motor could be described as efferent, going towards the effector organs from the CNS. One good way to keep these straight, it's tricky because they sound almost identical, is the acronym SAME. S-A-M-E, right? Where S-A puts together sensory afferent, M-E puts together motor and efferent. It's a good one to remember because, hey, these words sound the same. Let's use the word same to keep them straight. Neuroglia are the support cells. They are the pit crew or the stagehands. They are unable to manipulate ions to conduct action potentials, but they have lots of other uses. They are smaller but more numerous than neurons, so they end up comprising more of nervous tissue than the neurons do. They can't divide to produce more cells, so the healing that occurs here in nervous tissue is usually a function of the neuroglia. Neuroglia are going to be quite different in structure and function. That gives us really 6, but we'll talk mostly about 5 types. Astrocytes, ependymal cells, microglia, and oligodendrocytes are found in the central nervous system, whereas Schwann cells and another cell called the satellite cell is found in the peripheral nervous system. So most of the ones are in the central nervous system. Astrocytes are called astro for their star shape. They have many processes coming off of a cell body. These are the major supportive cells of the central nervous system, providing physical support for neurons as well as connections between neurons. They are capable of repair, so they'll form the scar tissue of the brain and the cord. They can stimulate and inhibit nearby neuron activity as well. Finally, an important function is the formation of the BBB, or blood-brain barrier in this case. The processes of astrocytes, you see here, will extend to wrap around nearby vessels. And by doing so, the astrocytes create a barrier between anything in the blood that may hurt the nervous tissue. This is a major protective structure, but it also makes it more difficult to create pharmaceuticals that can reach the tissue of the brain and the cord at therapeutic levels. Ependymal cells are the cells that line cavities in the brain called ventricles. Here they will produce a fluid called cerebrospinal fluid as they filter it from blood, and then with structures, cilia, they will circulate that cerebrospinal fluid all throughout the brain and the cord. This fluid protects and also aids in circulation of substances like nutrients and hormones. Ependymal cells, when you look at them, well, if you had to give them a tissue type, you might call them epithelial. And that kind of makes sense, right? Because epithelial cells function in secretion. They have microvilli and cilia. So maybe think ependymal epithelial, perhaps. And that might lead you to that secretion function. those microvilli will increase surface area for secretion, and then again the cilia can move to circulate. Microglia are the immune cells of the CNS. So they're eater cells. They will eat up bacteria and viruses, any cell tissue debris found in the central nervous system. Maybe think microbes are eaten by microglia. And then last one in the central nervous system is the oligodendrocyte. Oligodendrocyte kind of looks like an octopus, where it has this cell body-like area and then a few extensions that go out to the axons of nearby neurons, wrap around them, and form a structure called a myelin sheath. In the periphery, well, we can again see the satellite cells. They associate with the cell body and regulate passage into and out of the cell body, but you don't need to know those. You don't need to recall them for a test. The only one you need to know is the Schwann cell. Schwann cell wraps its whole self around a part of an axon in order to produce that same myelin sheath. Right? So both the central nervous system and the peripheral nervous system can have myelin associated with it, but it's a different cell that creates it, and it ends up looking different because of that. The myelin sheath in the peripheral nervous system is more robust because it's a whole cell that creates a portion of it. Yeah, myelin sheath, what the heck is it? Well, it is, like we just talked about, the multiple layers, up to 100 layers, that's formed around an axon by these neuroglia, depending on where we are, right? Central nervous system versus peripheral nervous system, the Schwann cell. This is a layer of protective wrapping that creates insulation around the axon, preventing the movement of ions. Because ions can't move through the myelin, it's going to limit where an action potential can form and greatly increase the speed and efficiency of an action potential. In a myelinated axon, the only area that an action potential can form is where there's no myelin. The naked nodes of Ranvier found between adjacent myelin. These are gaps in that sheath where ions can move across the membrane into and out of the cell. Because ions can move here, the action potential can develop here. Everywhere else where there's myelin, there's just so much cell membrane wrapped some more around the axon that there's no way that sodium can move in, potassium can move out there, and there's no way an action potential can form. So as we'll see in a myelinated axon, we'll see an action potential travel by way of saltatory conduction. Saltation refers to jumping, comes from the Latin for jumping. It refers to this jumping of action potentials that occurs from node to node to node, skipping all the myelinated bits, kind of like leapfrog. This is going to allow that action potential to travel faster, and it also cuts down on the energy that has to be used to maintain that resting membrane potential. So the efficiency goes up too. In addition, in the peripheral nervous system, where the Schwann cells are found, because the Schwann cell creates this really robust myelin sheath, it can also aid in regeneration of that axon. So neurons, they're unable to divide. So by and large, very limited capacity for regeneration and healing. The only place we can see healing of neurons is in the peripheral nervous system, where the Schwann cells are found. If a neuron has a myelinated axon and the cell body is still intact, that Schwann cell can help the axon regenerate. So, thanks to Schwann cells, we have a limited ability to heal our neurons. If you've heard of multiple sclerosis or MS, this is an autoimmune disease, meaning that it's our own immune system that is attacking healthy tissue. In this case, it's attacking the myelin sheath and the axon there. This is a degenerative, progressive disease that interferes with the neuron's ability to control our muscles. So over time, it leads to loss of muscle function. There is still no cure for MS, but ways to manage it have greatly improved over time. So now it's not nearly as scary and destructive as it used to be. And people that have it can expect to lead a much better life. This image shows the difference between a myelinated axon. This would be in the periphery, where we have multiple Schwann cells making up that sheath. With this crack right here, it says that node of Ranvier, where there's no myelin. It's very small, it's a very narrow space, but there's no myelin there, and we can see an action potential form, versus our unmyelinated axon, which there is Schwann cells there for support and protection, but the axon has all these uncovered areas, so an action potential will travel down the whole structure.